Hey guys, and today's video is going to be about indirect active transport in biology, and more specifically about sodium-glucose linked transporters and sodium-glucose linked transport, SGLT for short, because nobody's saying all of that all the time. And the two examples that I will be using are in the small intestine to absorb glucose, and in the kidneys to reabsorb glucose. And I haven't done a bio video in a while, so I hope you enjoy. So what is indirect active transport? Well, I think it's easier to show than to try to explain, but basically, there's no direct need for ATP for this type of transport, so it's technically passive. However, this type of transport would not be able to happen without direct transport being previously used. So that might seem a little bit confusing, convoluted, but I'll just show you exactly what I mean. And as I've said, our two examples are the small intestine to absorb glucose and the kidneys to reabsorb glucose. So let's look at the components of what we have in the small intestines. We have our epithelial cells, so just the cells that line our intestine, which are full with membrane proteins. The same thing can be said for the kidneys, which have, as part of their nephron, the nephron is a unit of the kidney which focuses on reabsorbing useful materials. It also has epithelial cells, which also have membrane proteins. And outside of those cells is the lumen, and this is where the glucose flows, or not only the glucose, other materials. We're just focusing on the glucose right now. And the goal of the small intestine is for the epithelial cell to absorb the glucose from the lumen and then into the blood. And in this case, glucose is going against the concentration gradient because we want to absorb all of the glucose, which, again, goes against the concentration gradient. So at one point, we'll be pumping in glucose into our cells when there's already a higher concentration of glucose in our cells than in the lumen. And the goal of the kidneys is pretty much the same, except we're reabsorbing the glucose, again, back into the epithelial cell and then into our blood. And the step to indirect active transport are as follows. So first, because we have all these membrane proteins, we have previously used our sodium-potassium pump to pump sodium ions into the extracellular side, so the lumen. And if you're not familiar with the sodium-potassium pump, I highly recommend you watch a video about that and figure out what that is before, because it's very crucial for this video. But just short and simple, it's a pump that transports sodium out of the cell, potassium into the cell, and it uses ATP. So this is an active type of transport. Now, if we pumped all of the sodium out of our cells, what we'll want to do? Well, sodium will want to go down the concentration gradient, so it will want to travel back into our cells. Now, in these epithelial cells, there are protein channels that allow for 2 sodium ions and 1 glucose to pass at the same time. So in order for this protein channel to be activated and actually open so that the components travel into our epithelial cell, we need 2 sodium ions and 1 glucose. And you need to have all 3, otherwise the channel doesn't open. So because the sodium wants to go down its concentration gradient, which is passive, it pushes the glucose into this protein channel and into the active site of the protein channel. This leads to the fact that the channel opens and both pass. And notice how we didn't really use any ATP for the actual protein channel that transported glucose into the cell. However, we needed that sodium to push glucose into the channel, and the only way that we got sodium to push glucose into that channel is to pump sodium out in the first place, which used ATP. Now, if this is still confusing, I've got a diagram for you right here. So let's look at our first step, which is right here. We are first pumping sodium out of the cell. And this creates a very high concentration of sodium ions in our extracellular side, so our lumen. This is our lumen. And the inside here is the inside of our epithelial cell. Sorry, that's very non-readable, but I hope you understand. So now sodium ions want to passively move down their concentration gradient So from a high concentration of sodium ions to a low concentration of sodium ions, as we can see here. And I've drawn squares over here to make the visual representations easier, because this is our protein channel that will act as a transport mechanism for the sodium and the glucose. The glucose are the little circles right here. So sodium ions want to get into this channel because they want to move into the cell, and in doing so, they push the neighboring glucose molecule also into the channel. And once there have been 2 sodium ions fitted in and 1 glucose fitted in, the channel turns and releases these molecules so that now we have transported both our sodiums and our glucose into the cell. And in this case, sodium moved down its concentration gradient, but glucose moved against its concentration gradient. So this is how it works. And again, this has required ATP, but here there was no ATP at all. So in that sense, it's indirect active transport, because we did need ATP in the step before, but technically this channel does not require ATP. And just as a last note, I would like to add that this type of channel is a contransporter. And what is a contransporter? Well, it's a type of transporter where one molecule moves down its concentration gradient while the other molecule moves against its concentration gradient. And this is exactly what we have right here. Sodium ions move down their concentration gradient, but glucose moves against its concentration gradient. So this protein channel right here is a contransporter. So I hope you're a bit more familiar with sodium glucose link transport and what indirect active transport is as a concept. And I hope this visualization really helped you understand. I hope you enjoyed, and let me know if you want any more biology videos. See you next week.